So it's really a privilege to be able to represent the field of epigenomics and our group. I do want to convey us a little bit as part of the source of the problem here that is really looking for a solution. If one looks at the Human Genome Project and its impact, literally nothing in the lab goes on today without using that genome sequence. And that's been really enabled by computational tools such as the UCSC genome browser. I would argue that the epigenomic maps and data that are being produced by our colleagues probably are going to be equally useful for the labs. And I'm going to show you today some things that we've been able to do with them. But I think that our field has largely failed in developing the interfaces and tools to allow the entire community to use this data. We are solicited constantly by requests from our colleagues to help them interpret these maps, and when we do, they find very useful things. But this is not a scalable operation. So for my talk, I'm going to give you a background on the epigenome and some of the features. How is DNA organized? I'll talk about mapping technologies, and then I'll give you sort of two vignettes from some of these ongoing large community projects. A vignette about how the developmental specification of cells is accompanied by a reorganization of the genome in structures that help commit cells to specific lineages. And I'll then talk about a very recent story where epigenomic maps have been useful for annotating and making regulatory predictions for the dark matter, for the non-coding portions of the genome that are really illuminating entirely new frontiers. And this as well, I think, will impress upon you why this is going to be so useful for the community of biologists and disease researchers. So as many of you are familiar, the fundamental problem here is that there are about 3 billion bases of DNA. This is 2 meters, it's about 2 meters in length, and it needs to fit into a nucleus that's about 1/10 the size of a head of a pin, right? So there are these layers of organization where your DNA is first wrapped around histone proteins to form these nucleosomes. This is about 150 bases of DNA. The nucleosomes then form higher order structures that remain poorly understood and then form into chromosomes and organizations that we'll hear a little bit more about in the following talk by Erez Lieberman. The structure of the genome, the epigenome, is not static. It varies greatly across the genome. These are just sort of very classical images showing that transcribed genes, region where there's transcribed genes are rather open. The chromatin sort of unfolds and gives the protein machinery access to the transcription, to the DNA itself. One can see this in other ways. So really, genes and control elements are accessible. I'll show you, though, we can gain far greater insight even into their functions by studying their structures. This higher level of detail, we heard a little bit about it in the last talk, but the DNA is directly modified by methylation. This is distinct from the A's, G's, and T's, right? This is another layer where you can actually directly modify the DNA. The histone proteins, this organization, these spools are also subject to dozens or maybe more than 100 different chemical modifications that really are signatures of specific functions. Different elements in the genome, genes, promoters, as you will see, have very different patterns in the modifications, and this is enormously useful for people studying the genome or genes or cell types. Epigenetics, there's really, just a quick note on epigenetics. Epigenetics really relates to the study of development and lineage specification. How does one single genome, which is sort of the title of this session, lead to such different phenotypes during development? These different cell types in your body that all have essentially the same genetic information have, markedly different phenotypes. The basis of that, we know, is that different genes are expressed in different cell types, and this is intimately related to how the DNA is organized, which is really the topic of this talk. I don't want to spend much time on this talk, but just understand this is incredibly important to the NIH. Epigenomics is central to human disease, it's central to cancer, it's central to neuropsychiatric disease and metabolic disease. We need to annotate, epigenomes, annotate the genome and the epigenomes to understand, the relationship between genotype and phenotype. And for this purpose, we need reference epigenomes, which we got covered. We're making a lot of data. Toolkits for analyzing epigenetic features, which we've got covered. and portals and interfaces, which I think is something that I'm looking to this community here for assistance in helping to drive this out into the community so disease researchers can use this data. So an overview of the technologies, just briefly. DNA methylation can now be read out in a whole genome scale by sulfite sequencing. I'm not going to talk a lot about DNA methylation today. DNA accessibility, we saw some data in the last talk, can be mapped by DNA-seq. And histone modification patterns, which you'll hear a lot about in my talk, can be mapped by ChIP-seq, right? So these are all methods that link conventional assays that have been in existence for many, many years with these new sequencing technologies that were just introduced in the last couple. Okay, so how do we map things across the genome? What is the underlying technology? Well, it's a cellular and biochemical assay. You actually take cells, living cells, and you fix them with formaldehyde. You take the chromatin, and the chromatin consists of these proteins, these histones in the DNA, and you use antibodies to pull out chromatin having particular structures. So you might pull out open chromatin using an antibody that recognizes a lysine 4 methylated histone. So you enrich these portions of the genome, right? You pull out the histones, but with them come the associated DNA. So then you can deep sequence this DNA, and you're not so much interested in the actual sequence itself. You're really counting how often you sequence different portions of the genome. And we saw in the last talk that these promoters get sequenced again and again and again in these assays, and that's because these promoters are methylated at this site, and the DNA gets pulled out in those immunoprecipitation assays, in those enrichment assays. This is kind of what the data looks like. The x-axis now is always the genome, and I'll just give you a sort of a classic depiction. You know, the epigenome varies by cell type, so let's start with embryonic stem cells, which are pluripotent. X-axis is the genome. Here we see a view from the UCSC genome browser of the PAX5, which is a master developmental regulator gene. It's in ES cells, like many other developmental genes, has this very characteristic chromatin structure where you see that you not only enrich when you do an IP for K4 methylated histones, so this is the activating mark, but you also see enrichment when you do an IP for the lysine 27 methylated histones, which was more of a repressive mark. So this is sort of a bivalent, we call it, signature with these opposing activities, both repressive and active. The gene is actually off because you don't see any of this transcription mark, but we think it's poised. This is a characteristic signature that tells us in this given cell, this gene can come on later. So this is enormously useful information for biologists and studying stem cell development or regenerative medicine. And we can now watch across different cell types, such as now we can look at hematopoietic stem or progenitor cells, and you can see you still have a bivalent structure, not so much change. But if you look at B cells, now where PAX5 is expressed, all of a sudden you see this switch. You lose the repressive mark, the activating promoter mark intensifies, and now there's this new mark of K36 methylation that's extending across the genome, signifying transcription. So really, all of these activities are marked at the level of the genome in terms of its chromatin, and they provide a very valuable tool for us to now not only annotate genes and other regulatory elements, but understand their activities in different cell types. So here comes the problem. We have now looked at something between 50 to 100 cell types just in our own group. We have looked at dozens of histone modifications. Our colleagues at UCSD and Bing Ren's lab have maybe looked at 100 histone modifications. And when you start pulling out the matrix here of cell types by different chromatin marks, the complexity is enormous. I'm going to show you that there's a lot of information here and we need to get at it, and we have some ways to get at it in the lab. But I think people are starting to see that maybe this is approaching some of the level of complexity where we're hearing in the keynote talk about other networks earlier today. So here are just data looking at each box here represents a collection of cell types. Each narrow stripe that you can see within the box is a single cell type. We've mapped lysine 4 methylation and activating mark sitting at promoters. Here's a mark that lights up regulatory elements, the transcript mark, the repressor mark. We can look at sort of correlations between the marks itself, but of course this sort of ignores the complexity and the details about all these individual cells and what they're doing. We can spend a lot of time sort of scanning across here the genes, right? We can look at each individual genes, but here you can see the complexity is already, it's a megabase of DNA, and the genome is, thousands of times larger than that. So, this is quite a challenge in itself. I should say that this is data from the Jiangsu and Mazar Adli, Noam Shores, Chuck Epstein, and colleagues in the Broad Epigenome Center. So here's one stab at taking a look at how to handle these data. One is you can sort of ignore the individual genes and the locations, but you can sort of identify principal components across the genome based on the landscapes of these different modifications across the genome. And I think it makes a compelling case that there's some order to these data. We can look at different types of cells, like stem cells, pluripotent cells, or blood cells, or tissues. And what we can see is for each of these principal components, the cells that are alike phenotypically cluster together quite nicely based on sort of an unbiased analysis of the principal components of the epigenomic landscapes. One can see this for each of the different marks. One can then follow up on the principal components and trace these components and ask, well, what is the basis of this difference? In many ways, it makes sense. This is that lysine 36 principal components associated with transcripts in these transcriptional of genes. And in fact, you can see that what we're picking up here that's distinguishing these different PCAs is different patterns of expression of genes that are common by these different clusters. So here's just sort of a, for example, in some of these blood and immune cells, one sees immune response genes are activated and making a K36 marks and making them all sit down here. In brain tissues here, one can see high activity of neuron-related genes, right? So this is a basis of this clustering. We can also, though, I think one of the special values of the epigenomic maps that can't be obtained from gene expression analysis is that there are other modifications that light up distal elements like enhancers, right? So the genes encode only a few percent, cover only a few percent of the genome, but this other 95% of the genome is actually incredibly important as well. And this is where the regulatory elements lie, and how do we identify them? K4ME1 is a powerful means for this, and we can see sort of a complexity in these regulatory elements and when they have this chromatin structure. And I'm not going to spend time here on this because I'm going to come back in the end of the talk. What about developmental specification, looking at ES cells versus which are pluripotent versus all other cells which are really differentiated? There's a striking difference in the K27ME3. This is a repressive chromatin mark. And what I think one can see, just taking, this is easy to see, in looking at ES cells, you see very sharp peaks of K27, but in differentiated cells, the peaks are much broader. This reflects sort of a large-scale reorganization, and we think a compaction of the genome that occurs during development. We can actually observe this occurring as you differentiate ES cells into neurons. There's a compaction scale that Zhang and Mazar Adli have developed that shows this increasing compaction of the chromatin, increasing spreading of the K27. And as this is sort of a artist's depiction here of the nucleosomes compacting, and we can see a loss of some of the activating marks as this occurs. And it's very tempting to conclude that this helps restrict lineage as cells differentiate, that they cannot now switch into other cell types. So for the last part of the talk then, I want to talk about some work that's this group I've mentioned has been involved in, but in particular has been an intense collaboration with Jason Ernst and Manolis Kellis in the computer science department at MIT. So there's data collected in the context of the ENCODE project now, where we've looked at about 10 different modifications in a compendium across about 10 different cell types. And what we want to do here is not only find genes, but we want to identify distal elements such as enhancers. And we gain power doing this now by looking at combinations of histone modifications at the same time, not just looking at K4ME1 maps, but looking at all 10 maps at once. And what Jason and Manolis were able to do is use a hidden Markov model to do a couple things. One is to scan across the genome in a lot of different cell types and learn recurrent modification patterns or chromatin states that are sort of combinations of chromatin marks or histone marks that come together. Then they were able to annotate the genome according to these different chromatin states. And if they then look at the chromatin states, these were learned really in an unbiased, sort of purely computational way. But as we look at the states, we realize for a number of reasons that these states actually correspond to different kinds of promoters. They're near to transcription start sites. They're DNA sensitive. They have transcription factors bound and so on. Other states tend to be farther away from promoters, but they look like enhancers. They have a lot of transcription factors bound and so on. Other states are these transcribed states and so on. And then there's, of course, the heterochromatic repressed states. The attraction of this is we've now been able to take about 180 data sets and something like 100 billion bases of DNA and collapse it into a single genome-wide annotation for each cell type that is an annotation based on these different colors of chromatin states that we argue actually contains most of the information that we want to get at here. So we can now, instead of having to have many tracks, 100 tracks on the genome browser, we can actually just have one track per cell type. You can see these different colors lighting up. Here's a promoter of the Wintless gene. It's purple or poised in ES cells. There's an active signal in these differentiated primary cells and so on. And now, it's important, I'm going to talk a little bit about the enhancers of the distal elements, and I just want to point out that we've validated that these are we're really picking out real enhancers by these methods because we can go back into the cell and we can show that these sequences truly enhance transcription. So we wanted to begin to annotate these enhancers, these distal elements, and sort of incorporate them into a genome regulatory network. Right? We've seen a lot of these networks that connect genes. But if you think about the underlying biology and the mechanism, genes connect to each other via regulatory elements, via transcription factors and regulatory elements such as enhancers that then activate other genes. So we wanted to incorporate these transcription factors and these enhancer elements into this regulatory network. What did we do? Well, first we looked at the different patterns of activity for promoters across different cell types. We can see in sensible ways that promoters associated with K562, these blood cells, erythrocytic cells, have gas transport genes associated with active promoters in immune cells associated with the immune response. So we're doing something right. We can also annotate enhancers and their cell type specific patterns across different cells. They're exquisitely tissue specific. These enhancers are not static at all. If you look in different cell types, you can pick up based on the chromatin marks which cell types which enhancers are on and which cell types. And again, now you can take an enhancer, you can look at nearby genes. So enhancers that are on in immune cells, if you look at the nearby genes, they have immune functions. Enhancers that are on in liver cells, if you look at nearby genes, they're involved in lipid metabolism. So this is making a lot of sense, suggesting that we're picking up the enhancers and we're beginning to identify the nearby genes they may be regulating. We can use fancy correlations to link enhancers not only to the nearest genes, but to farther away genes based on common activity patterns, right? You can look for the pattern of activity of enhancer, and you look for a nearby gene that has a similar activity pattern across cell types. So now we can build a network that has got a layer of connectivity, not just identifying the enhancers, not just annotating their cell type specificity, but now making predictions about their gene targets. So I think we could see that this would be a nice capability to build into some of the genome browser and other tools. And I didn't show you, but we have some reason to believe that this is relatively accurate based on other confirmations. But I wanted to jump quickly and tell you that we can also predict the upstream transcription factors, right? We saw some of the browser snapshots showing conservation, and these are ways people can identify transcription factor binding sites. So we can use some of this data to identify the transcription factor binding sites that are enriched in specific enhancers. And we can now develop a very rich and complex views that I would hope could be made more accessible, but I think at this point are pretty complicated, but very rich in their information content, describing these different enhancer clusters. So remember I showed you before there was a cluster of enhancers at the top that was active in ES cells. It turns out if you look at the motifs, you see that those enhancers have motifs for OCT4, which is, that's shown here in red, but that OCT4 is also specifically expressed in ES cells, shown here in yellow. So this is basically, when you see red and yellow next to each other, you've got an inkling that here you have a factor that is whose motif that may target these enhancers and who's specifically active in cell types where these enhancers are on, thus suggesting this is the factor that's activating these enhancers. We can see a similar thing for this sort of liver-specific cluster of enhancers. And if you go over here next to HepG2, you can see this sort of cluster of yellow and red, and they correspond to H and F, which are really well-described liver transcription factors. So these are liver-specific enhancers that seem to be driven by H and F factors. And there's many other examples as well across here, but I don't have time to go through all the details. And you know, we can validate this in experimentally validate this by doing, again, enhancer assays in cells. So just to sort of wrap up this network model, we've taken sort of a classical gene network model, and now we've begun to build into it these distal regulatory elements. We've linked those elements to genes. We're predicting transcription factors that may be regulating these enhancers, and we now have a rich model for annotating the non-coding or the dark matter of the genome. Why is this so useful? Well, I'll give you one example. I think many are familiar with the genome-wide association studies that are now beginning to identify the sequence variants in the genome that are associated with human disease. So, right, there are many variants in these SNPs across the genome that have been cataloged. And if you look at just your average run-of-the-mill SNPs, they don't show any predisposition to sit in any particular chromatin environment. But if you look at SNPs that have been associated with disease, all of a sudden you see an enrichment for the enhancer elements that I've been talking about and showing you. So SNPs associated with disease have more than a twofold enrichment to sit in chromatin regulatory elements that look like enhancers, okay? So, but it goes even a little further than this, because now if you ask what are the specific diseases, and you zoom in on some of the specific data sets from the GWAS catalog, you see that there are specific associations between diseases and enhancers. So SNPs, or at least phenotypes, so SNPs associated with erythrocytic phenotypes correlate to enhancers that are specifically active in erythrocytic leukemia cells or K562 cells. So these SNPs are sitting in enhancers that are active in the right cell type. SNPs associated with arthritis, cirrhosis, or with systemic lupus erythematosus, these are immune-related diseases. These SNPs are sitting in enhancers that are specifically active in the immune cells we studied. And again, you can see lipid diseases, lipid phenotypes, the SNPs are sitting in HepG2 specific enhancers. So this is suggesting a couple things. One is it's helping to affirm some of the SNP results and the associations from the disease phenotypes. It's beginning to identify models where these might be precisely studied, and it's also suggesting that the chromatin-based annotations can be very valuable to a whole community of researchers who are studying variants and trying to understand their role in disease. This is just showing you all, there's a lot of SNPs. These are the annotated states for each of the SNPs. And blood lipids, you can see under the liver, there's a lot of enhancer structure under the SNPs. And it's specific to HepG2. Erythrocytic phenotypes, again, in the erythrocytic cells, you see a lot of enhancer structure sitting under the SNPs that have been associated with erythrocyte phenotypes. How might this be useful? Well, now we're back to the genome browser, and we can look across these regions where some of the associated SNPs. So here's a SNP from the blood lipids phenotype. The red is the lead SNP, but any of these SNPs, all of these SNPs are sort of were associated in the study. And one might argue that this associated variant, you might triage this one for further study, because this may be a causal variant. Why? Because it's actually sitting right inside of an enhancer that's specific to the right cell type, HepG2 cells. Another example is an erythrocyte phenotype SNP that is sitting right next to a K562 or erythroid cell-specific enhancer that actually affects one of those transcription factor motifs that we predicted to be regulating the enhancer element. So this might be this is a hypothesis about which SNP might be causal and how it may be acting. And there's other examples, such as looking at lupus and a SNP or a variant that's affecting a binding site for ETS1, which is another one of our predicted regulators. So just for my conclusion then, I think I've shown you a lot of data, but I hope at least I've conveyed that these epigenomic data sets have great value for annotating the genome, in particular the non-coding genome, which has been rather inaccessible thus far, and they can provide insight into regulation across cell types and developmental stages. This data is a great resource for the biomedical community, but clearly we need to improve the accessibility and you know, whether it's literally three-dimensional views, but at the very least, browsers that incorporate additional dimensions of the data, such as connectivities, transcription factor motifs, and so on, I think are essential. And finally, I can tell you that, you know, I'm part of these large consortia, including the ENCODE consortia and Epigenomics consortia, and we have, you know, dozens of colleagues around the country and initiatives in Europe that are now mapping beyond just these chromatin marks hundreds of different transcription factors, non-coding RNAs of all different types, looking across many in vivo tissues. This is going to be a great resource. This is really going to change how we can study human disease. And it's going to be something that the whole community wants to get at. So I just should thank my colleagues at MGH Pathology, especially Mazhar Adli and Zhang Zhu here, and James Zhao as well in this project, as well as at the Broad Institute. We have a fantastic epigenomics team that is pouring out this fire hose of data and doing some really exciting technology development and computational analysis. And of course, I've mentioned Manol Kellis and Jason Ernst, MIT, and our colleagues at Harvard, including Alex Meisner and Chad Cowan. Thank you very much.